So, hello everyone. Thank you for joining us today. I am Annabelle, Solis BioDyne's product manager, and we will be introducing our next generation reverse transcriptases for you. If you have any questions, meanwhile, please, you can write them down in the chat, or if something interests you, then also just add it to the chat, and I will look these either questions or ideas. I will look them over at the end of today's webinar. So we have a little Q&A coming. So now the introduction is done. So the new products of ours are FIREScript ULTRA H+ kit and FIREScript FLEX H- kit, as you might have already seen from our teasers or our posts overall. So you might wonder what this H+ or H- actually stands for. So in case of H+, it refers to the full RNase H activity. Therefore, this plus is included in the name. And with the H-, it stands for the reduced RNase H activity, or I would say even that it's not detected with this FIREScript FLEX reverse transcriptase. Moving on, when Ultra H+ has this full RNase H activity and H- has reduced, here is also the representation of the activity of RNase H. So in this case, we have taken FIREScript, our old generation FIREScript, as the reference. So you can see that this Ultra H+ is over 20 times more active in terms of RNase H. And in this case, this activity can improve sensitivity in certain applications, thanks to removing the RNA from this RNA-cDNA hybrid. And when you see down here, you can also see FIREScript FLEX H-, which is similar to the non-template control in terms of the activity, which we can think of as the not detected RNase H activity, therefore not having any activity whatsoever. And what is more about this RNase H activity and having it with the enzyme, there is more single-stranded DNA available during the first cycles of the PCR when you are moving on from the cDNA synthesis to downstream PCR. Now, about the key features of the new products would first and foremost be the wide working temperature. So both of the enzymes work at a relatively wide working or wide temperature range, as you can see on the figure on the right. This experiment was done for 10 minutes, and you can see that in case of Flex and Ultra, they can do effective cDNA synthesis and afterwards PCR really well in a long incubation of the cDNA synthesis. So even up to 70 degrees Celsius. When we think about more about in terms of optimal temperatures, then on the left figure here, I have pointed out the, so to say, optimal temperatures for the reactions that can come out in shorter reaction times. So in this case, you can definitely say that our products work really well with standard, so 50 degrees Celsius temperature as well, as shown on the picture on the right. However, if you want even more beautiful or amazing results, then you can think of trying or using our products with a bit higher temperatures. So around 60 or 65, which is even beneficial for some applications that require higher the usage of higher temperatures. Now, as I already mentioned a little bit about speed, the products of ours, so the Ultra H plus and Flex H minus, both work really fast. Here is just a representation of the reactions done at 5 and 10 minutes, but they are done at 50 degrees. So we can actually also tell you that the synthesis can work even faster. So if the conditions are optimal, so the temperature is a little bit higher, then you can even do the reactions in less than 3 minutes. So in 1 minute or 3 minutes as well. And about the possibility to do or synthesize cDNA in terms of length, we have done the experiment with over 12 kilo base pairs of cDNA. So you can definitely say that our products, they can do this longer templates really well. We haven't tested longer cDNAs yet. However, there is high chance that they work well even with longer cDNAs, or for longer cDNA synthesis, so with longer RNAs. In this case, both of these experiments were done at 50 degrees. So as I mentioned before, this might not be always the most optimal, but it shows that our products work even at the standard conditions and can work even more better with higher or more optimal temperatures. Now moving on to sensitivity. With Ultra H plus, we have seen that it as it removes the RNA from the hybrid, and there is more single-stranded DNA in the first cycles of PCR. Then we have seen that it also is shown or seen in case of qPCR and the cycle threshold values. So the amplification curves, they come up earlier than it is with the competitors. Also about the stability of our products, as you might know that we have promised, or at least we know that our products work really well at room temperature for at least a month, or for up to a month. However, in this case, we also tested incubation at 37 degrees for a month, which is shown in red, and the reference is shown in blue. So you can see from here as well that in case of both products, both Flex and Ultra, these amplification curves of reference and the one that is incubated at 37 degrees for 4 weeks, these amplification curves come up really, really nicely, and there is minimal to no change in the functionality of the enzyme. What is more, the enzymes, as you have understood probably from the previous slides, the enzymes are really, really heat tolerant, or they work at high temperatures. So they also have tolerance to different treatments, such as a heat treatment beforehand. So in this case, in this experiment, we tested our Flex H minus enzyme and compared it to our old generation FIREScript and one of the competitors' products. With this light color, you can see just the standard conditions. So no pre-treatment, just the reaction itself was run. And with the darker colors, there was pre-treatment of 65 degrees for 30 minutes done before the actual reaction. So in case of FLEX H-, this change in cycle threshold was really minimal. So just somewhere approximately one cycle or one cycle threshold. Whereas with other products, there was a way a bigger change in the effectiveness of the enzyme. So you can definitely be sure that our product, this FLEX H-, whenever you have a certain application that requires pre-treatment or at higher temperatures, or there is overall a higher or high temperature usage before this cDNA step or cDNA synthesis step, then you know that this FLEX works really nicely, and it can tolerate the heat shock or heat treatment really well. What is more, I will now talk to you a little about another really amazing feature of our products. So this is benchtop stability. To give you a little insight, what we mean by the benchtop stability is that when you mix all of the products, all of the components of a cDNA synthesis reaction together, this includes also RNA and primers and DNTPs. When you mix them all together into the tubes, then when you leave them at room temperature overnight or for 24 hours, then the stability actually is there. So there is no to minimal change in the reactions afterwards. So this is actually really good when you're accidentally leaving the pre-mixed reaction on the shelf, or you just want to leave it overnight because maybe it's already pretty late, or in case of any like high throughput workflows, that when you have a lot of reactions and you have to leave them for a long time already pre-mixed, then you can be or rest assured that the products or the reaction itself does not start before you actually want it to start. And this is actually thanks to our enzymes inherent warm start. And this is not that we have added this warm start, but this is rather the performance that these new reverse transcriptases themselves have. So they don't have it, but at the same time they can give similar performance to as if it had this warm start. So we would say that it has inherent warm start. And to show you what actually happens, or how this is proved by us, then on these upper figures on A and C is our FLEX H- product. So in this case, the amplification curves and the melt curve. And on the figures below, there is the competitor's reverse transcriptase. And you can definitely see that with these green and blue amplification curves, these are just the standard conditions. So when the reaction was not incubated before the run, so it was just mixed and put into the machine right away. And with these pink and gray ones, these are the ones that were incubated at room temperature for 24 hours before this reaction itself was run. So in case of competitor's product, you can see a lot of unspecificity happening. There is the product that we don't have or don't want to have. And you can see it also from this amplification curves. They come up really early. However, with our case, they come up when you want them to come up, and the product itself is also correct. So, and this is, again, thanks to this inherent warm start that the RTs have, that our RTs have. And it is really essential for high throughput workflows. So whenever you're thinking about doing these types of workflows or have applications that require good and precise timing when the reaction starts, then you can be sure that our product actually works and might be of benefit, might be beneficial for you. Moving on from the stability, our RTs also have good tolerance to common inhibitors. So here on the screen, you can now see some of them, some of them that we have tested. So different inhibitors from soil, blood, and urine. Our enzymes, both of them have good or even great inhibitor tolerance to them. Now, and to show you as an example with this FIREScript FLEX H- case, we tested out one a bit complex target gene and with these inhibitors that I showed you before as well. And you can see that this CT shift, or the difference when having this inhibitor and not having this inhibitor present in the reaction, there is actually minimal change in the sensitivity of the reaction. So this shows that even if there is some inhibitors present, the impact of the inhibitor to the enzymes is minimal. Now, I covered the most important key features of the products, so both of the ULTRA H+ and FLEX H-. Here you can see the catalog numbers that you can use to either find it or search it on our website, or to tell our salespeople that you want to get one or the other product. Now, we have different formats for these products. So the sample size is 20 reactions. So you can get this sample after our webinar. You can definitely let us know that you would be interested in having one or both of them. Then, additionally to the sample size, we have 50 reactions and 200 reactions as catalog formats. However, if you want to purchase bigger reaction sizes or do bulk size purchases from us, then this is also definitely possible. Now you see that we have in both cases we have 3 vials in this kit. So these consist of the enzyme, so either ULTRA H+ enzyme or the FLEX H- enzyme. Then there is the advanced reaction buffer for that is optimal or optimized for these enzymes especially. And we have decided also to add our RNase inhibitor RiboGrip into these kits. And if you have seen our other products before, then you might wonder that but you have RNase inhibitor added to this, so to say, smaller kit. Why is that? So, as you might know, RNA is really unstable, and there are a lot of RNases that want to degrade the RNA. So it's really important for us to think about the protection of the RNA. And the biggest, so to say, bad guys here are the RNase A, B, and C. And in this case, our RiboGrip is really, really essential to be used. Together with the glycerol version of our RiboGrip, we also have glycerol-free version of this product, and they are both essential to be used as protection from the, so to say, bad RNases. They're really important for the RNA-based applications, not only cDNA synthesis, but also the other different applications that might require RNA as the initial template. And as you might know, in case of RNase A, it wants to degrade this single-stranded RNA, and our RiboGrip, it quite literally crypts it, so it doesn't let it go and degrade the RNA. So really essential product to have as complementary product to our RTs overall. Now, you might be interested in also about the application areas. So you're definitely from a lot of different fields and labs or distributors that are here. So our products work really well in different applications, definitely. So whenever you're working with different gene expressions, analyses such as RNA sequencing, library preparations, single cell sequencing, etc. For example, then it might be really good. These might be really good products for you. As well as when you're working with different target discoveries or need to go into more mRNA or biomarker detection and research, then also. When you're working with infectious diseases or overall viral RNA, then also for these cases, our products are really, really good. So as you can see from this slide, a lot of different aspects and application areas can be covered with our products. So whichever area are you from, you can definitely find something or find either Ultra H+ or Flex H- beneficial for you. So to so to say conclude more or less this webinar, I would like to go over again really important keywords that you should, so to say, take home with you or take into your day. So in case of them both, so with Ultra H+ and Flex H-, you can think of them as having this inherent warm start, definitely, which gives both of them really, really good other features, such as this benchtop stability that I talked about, overall great stability, so at room temperature, which is really good because we can send them out without any ice or blue ice. We can send them out in envelopes, so they're really convenient for you. Also, they have great inhibitor tolerance. So if you're working with more complex samples overall that need that are inhibitor rich or that overall need a bit higher temperatures, then our products are really good in that case also. Also, the products are the revertases are really fast. So if you need to the cDNA synthesis to be done really quickly, then you can be sure that our products do it very well. And also, when you need to do full-length cDNA synthesis, then you can rest assured that definitely this Flex and even Ultra H+ can work really well for you. About Ultra H+, more or less, it has higher sensitivity, or it can give you quicker amplification overall in qPCR later on. It is good for targeted RNA detection wherever you need it to come up, needs amplification afterwards to come up quicker. And when you need the reaction itself to be, I would say, more clean, or like where you don't have this hybrid situation happening. And in case of Flex H- product, it has high persistivity, so it can do really the full-length cDNA synthesis. It has a great tolerance to heat treatment, and it has already been used in single cell sequencing. So whenever, or if you're working with single cell sequencing as well, then, or any other type of RNA sequencing, then these products can be used in those cases as well. Now, as you might know, our portfolio is pretty large already. So these products, they are under our reverse transcriptases, as shown here. So if you think that we have only those reverse transcriptases, then you might be wrong. So if you want to have like a full set of different, for different applications, such as maybe you need the reverse transcriptase as well as the afterwards PCR or qPCR products, then we also have those. So whatever you might need, you can ask us, and maybe you can already get everything from us for your workflows. Thank you all. I'm really thankful that you all could join us today and for the feedback that it was informative. So this is really good to hear. And yeah, please order samples if you want to try them out. They're really good. So thank you and goodbye.